Enzymes. Many of us have heard about them. Few of us have actually used them, and some may have seen great benefits from them. But what are these enzymes? How do they work? Are bacteria and enzymes the same thing? Can we substitute bacteria with enzymes or enzymes with bacteria? All this and much more in today's video about enzymes in wastewater treatment. Hello, my name is Sunil Budke here from Avalon Biotech. This is video number 3 from Avalon Biotech's series number 1, Fundamentals. In the last video, we talked about bacteria. We learned that a bacterial cell contains many chemicals and that thousands of chemical reactions are happening inside that tiny cell. Now, what happens in a chemical reaction? Two chemicals come together, bonds are made or broken, electrons may get exchanged or atoms may get swapped. The key point is bonds are either made or broken. And it is this making and many times breaking of the bond requires energy. Many reactions require external energy to proceed. This energy acts like a limitation, a barrier that prevents the reaction from happening spontaneously. If only there was something inside the cell to remove that barrier. Inside a bacterial cell, the temperature and pressure are fairly uniform. Yet, thousands of reactions are happening in the same tiny space. If all these reactions start happening at random, there will be chaos inside the cell. To control that chaos and to help these reactions occur efficiently, bacteria use enzymes. Enzymes act like catalysts in a chemical reaction. In simple words, they make it easier for the reaction to happen. Technically, they reduce the amount of energy required for the reaction to proceed. And by doing so, they accelerate it. Enzymes remove those barriers. Specific enzymes remove specific barriers. They are not part of the reaction itself. They are not consumed or changed in the process. After the reaction is finished or completed, the enzymes are free to participate in another similar reaction. So how big are these enzymes? Let's compare them with bacteria. A typical bacterium is about 1 to 2 micrometers, while enzymes are about 5 to 10 nanometers. That means a bacterium is roughly 1,000 times larger than an enzyme. But the real difference isn't just the size, it is a function. A bacterium is like a factory with different departments performing different functions towards a common goal. An enzyme, on the other hand, is like a tool used in that factory. It only has one specific function. Enzymes are highly specific. They act on only one particular chemical. If the chemical structure is changed even slightly, the enzyme will not work on it. Each bacterium produces 1,000 different types of enzymes. Some are released outside the cell, called extracellular enzymes, while others stay inside the cell, called intracellular enzymes. Which enzymes are made depends on the genes present in the bacterial DNA. When bacteria divide, the new cell make their own enzymes. So bacteria do not need any external enzymes for their survival or growth. All bacteria, and in fact all living things, make their own enzymes. Bacteria and fungi also continuously keep releasing these enzymes outside the cell into their environment wherever they detect food. The enzymes break down the large food molecules into smaller chunks, and then the cells are able to absorb those smaller molecules for energy and growth. Now, when we say that enzymes make the bond or enzymes break the bond, it will sound as if enzymes are alive or acting intentionally, like they have a mind of their own. But enzymes are not alive. They are not living things. They are simply large protein molecules. Enzymes do not grow, multiply, or reproduce. They simply act on specific reactants. Enzymes are nothing but proteins or protein complexes. Proteins basically are chains of amino acids that are folded together in a three-dimensional shape. Here is an image of an enzyme which shows that its surface has electrical charge pattern. This charge pattern helps the enzyme bind to its substrate. Let us now try to understand how enzymes work. I have made a model enzyme using Lego blocks. Imagine this Lego block is an enzyme, and here we have substrate. We want this enzyme to break the substrate into two parts. Now, many times students of biology are taught that enzymes and substrate fit together like a lock and key, something like this. But that is not correct. If substrate and enzyme would fit like a lock and key, then there would be no reaction. Instead, enzyme is shaped to match the transition state of the substrate being broken down. Something like this. Now, observe what happens when the enzyme binds with the substrate and the complementary charge distribution pattern tries to modify the substrate. Let me push this substrate into the enzyme and see what happens. You just saw how enzyme works. Please note that this is an oversimplification of the concept. The products are now free and the enzyme is free to work on other substrate molecules. Since enzymes are protein, they can get denatured, meaning they can lose shape. And shape, as we saw earlier, is very critical. If an enzyme loses its shape, it becomes inactive. Enzymes can get inactivated due to several reasons. Changes in pH, the temperature, the presence or absence of metal ions, or due to certain chemicals in the wastewater. Just a few months ago, a review was published in Science Direct. Enzyme sources in wastewater treatment, their influence on enzymatic bioremediation and large-scale application. This review summarizes research on enzymatic wastewater bioremediation published between 2014 and up to 2024, primarily sourced from the Science Direct database. It is a review of studies that directly employed enzymes for wastewater treatment for the last 10 years. So this is a very important paper if you are interested in using enzymes for bioremediation. Right in the beginning, the paper states that while commercial enzymes are available for various sources, high cost and stability issues remain significant barrier to large-scale application. Meaning, they work great in laboratory setup but fail when applied on a large scale. The effect of these commercial enzymes is short-lived. Why is it that the effect of pure enzymes is short-lived while enzymes produced by microorganisms exhibit exceptional stability and catalytic efficiency under harsh environmental conditions? Why does that happen? Let us take an example to make this clear. Suppose we want to break down cellulose, something very commonly found in the paper industry effluent. For this, we usually recommend our product called EvoBac in its powder form, which is designed specifically for cellulose degradation. But cellulose can also be degraded using an enzyme called cellulase. So why not use just that instead? Here is why. In laboratory condition where you have only cellulose in water, cellulase works beautifully under the right pH and temperature. But in an actual effluent where hundreds of other chemicals are present and the pH and temperature keep fluctuating, the enzymes become unstable and often ineffective. Complete degradation of any complex molecule requires multiple types of enzymes working in sequence. The enzyme cellulase is a complex that typically consists of three enzymes working together that happens naturally inside the bacterial cell. Inside the cell, enzymes are protected. The bacterial membrane maintains pH, filters which molecules can enter or leave, and keeps the environment ideal for enzymes to work. This protective control allows bacteria to keep their internal chemistry stable even when the external conditions in the effluent vary. We'll explore how this happens in detail in a separate video about transport inside and outside the bacterial cell. This video is crucial if you want to understand what causes toxicity to microbes and how to protect your system from shock loads. It will also help you maintain healthy MLSS level. So please don't miss that one. The research paper further states that microorganisms are a promising enzyme source but having some drawbacks. Although bacteria, fungi, and microalgae are feasible enzyme sources, the same enzyme produced from different microorganisms has varying performance. Therefore, for a specific industrial application, it is necessary to identify suitable microorganisms from a large database. And that's exactly what we do at Evlon Biotech. We have a large library of bacteria and we select the ones that are best for a specific application. Let me explain this further. Enzymes are proteins, which are long chains of amino acids. The shape of an enzyme depends on the sequence of those amino acids, and the sequence of those amino acids depends on the DNA. So different DNA means different enzymes and therefore different chemical reactions. So when we have two different bacteria, what we really have are two different sets of DNA producing two different types of enzyme systems. So even if two bacteria are producing the same enzyme, meaning enzymes to do the same thing, their enzymes can be very different in terms of their efficiency. All enzymes are not the same. I'm sharing a link to this research paper in the description below. Coming back, the most important thing to remember is enzymes do not reproduce or multiply. Bacteria do. Bacteria multiply. They multiply continuously and adapt to the effluent, improving their enzyme systems over the time. So does this mean that enzyme formulations have no use in wastewater treatment? No, not at all. Every technology has its own application and limitation, and enzymes are a great solution in many situations. Sometimes enzymes can be used to kickstart a reaction before bacteria start multiplying. We will discuss such cases in the application series, but such situations are very rare. For most practical purpose, bacteria is a sustainable solution than only standalone enzymes. At Evlon Biotech, our research over the last decade has helped us cover different effluent characteristic degradation profile, different enzyme complexes, and bacterial selection, ensuring each formulation is tuned for specific water conditions. Of course, this isn't a product video. We'll talk about them in a separate product showcase series. In the next video, we'll talk about how bacteria reduce COD and BOD in the ETP or STP system. Later, we'll see multiple videos on COD and BOD from video 9 onwards and multiple separate video on MLSS. This is Sunil Budke from Evlon Biotech, where we have bacteria for every wastewater treatment problem. Once again, thank you for your time. Keep learning, keep growing, and take care.